Today, I will be talking about the analytical and clinical validation of the breast cancer Oncotype DX recurrence score. This is a very exciting time right now in genomics. With next generation sequencing, there are multiple opportunities in screening to detect cancer earlier. And there are several companies, Thrive and Grail, that are offshoots of Illumina that are working on technologies to do that. Tonight, I'll be talking about the Oncotype DX recurrence score, which is used to help determine whether or not a patient needs chemotherapy that has hormone receptor positive breast cancer. But there are exciting applications of next generation sequencing for minimal residual disease to detect who has residual disease after chemotherapy versus who has been successfully cured by chemotherapy. And these are technologies that we're just starting to see in the clinics and presented at ASCO today. And of course, you're familiar with those patients who are unfortunate enough to have metastatic disease. And again, we're using next generation sequencing to identify genomic variants that can be targeted with targeted therapies. But tonight we're talking about the Oncotype DX recurrence score. And about 20 years ago, there was the recognition that many patients were being overtreated with chemotherapy. And this is a slide from the breast cancer overgroup. This shows that the benefit of first generation chemotherapy is just right around 6%. Many patients were cured by surgery alone, and some patients recurred even in spite of chemotherapy. But in the year 2000, the National Institutes of Health, the NIH, came out with a special article in which they had a challenge to identify women, specific patient populations for whom avoiding chemotherapy would be possible. And they recognized at that time that pathology factors, many of which I provide to my clinicians on a daily basis, were very limited with being able to answer this important question. We worked, first of all, to be able to develop a technology to help answer the question, who will benefit from chemotherapy? And first, we needed to make a decision in the platform what type of tissue input to use. And there was many folks that wanted to use frozen tissue. And being a pathologist, I argued that we must use formalin fixed tissue. It is universally available, it can be easily shipped, and it allows the pathologist to identify exactly where the tumor tissue is. Once we decided that we would use fixed paraffin embedded tissue, we needed to decide on a technology platform. And we decided to use quantitative RT-PCR. This enabled us to create primers that were very specific for genes of interest, in this case, mRNA or messenger RNA. And what PCR enables you to do is target a gene with great sensitivity. And PCR has a very wide dynamic range. It can measure several thousand-fold difference in gene expression. So when I read a ER stain using immunohistochemistry, I can tell you how many cells are roughly positive and what's the intensity. Is it 1, 2, or 3? But PCR can give you a wide dynamic range, 14 to 20,000 fold. And it's also very useful for working to measure mRNA from fixed tissue. Now, this figure right here, this Western, shows you that frozen tissue has very long and preserved mRNA fragments. Formalin fixed paraffin embedded tissue, even though the tissue fixes, the RNA continues to break down. But you can still see here, this 100 base marker shows you that many of these fragments have lengths of around 100 base pairs. And we did many studies to show that if we took samples in delayed fixation, or if we underfixed for a few hours or overfixed for many days, or if we used different fixatives, that PCR was able to quantitate gene expression or mRNA levels. And this is an example showing you samples that we fixed in a fixative called hollandaise, and then same tissue that we would fix in another fixative, formalin. And using RT-PCR in reference genes, we were able to control and adjust for delays in fixation for different fixatives. Today, most people are using neutral buffered formalin, but for many years, there were different fixatives. In reference normalization, and the recurrence score uses 5 reference genes, allows for compensation for these pre-analytic differences. Technology also, this is an experiment where we took multiple tumor blocks from a patient breast cancer sample. And we then cut sections that we stained with H&E and then adjacent sections, which we used for recurrence score testing. And we did this at multiple levels and across multiple blocks. And what we identified, and this is showing you a slide where the RNA gene expression is on the y-axis, and all of the plots I show you will show 2, 4, 6, 8. This is a log base 2 scale. So 2 to the 2 is 4 copies, 2 to the 3 is 8 copies, 2 to the 4 is 16 copies. This is after normalization. So a higher number means higher gene expression. So this is one of our normalization genes or reference genes, and you can see beta-actin is very highly expressed compared to this patient sample here. This is the ER expression, and it is much more lowly expressed. And the red and blue lines are showing you 2 different sections from the same block that have been tested separately. And you can see they almost look like a fingerprint. So this technology is very reproducible. The other thing that we noticed is that we needed to enrich for tumor tissue. And so we looked at what's the impact if you leave in skin, or what's the impact if you leave in Needlecore biopsy. This is a healing biopsy site that I am highlighting in the middle of a breast cancer because it was diagnosed with a Needlecore biopsy. And we wanted to understand what is the impact of a biopsy cavity on, in particular, the 16 genes that are in the recurrence score, because there is healing macrophages and proliferation in a healing biopsy cavity. And what this plot demonstrates for you is here on the y-axis is the recurrence score from patients who have whole sections, no dissection of the biopsy cavity. And here are patients that have dissection of their samples. And you can see on average, if we take one of these 53 cases, the pluses are well-differentiated tumors that were graded histologically. The 0 circles are poorly differentiated and the black X's are moderately. And you can see this well-differentiated tumor, if one left in the tumor cavity, they would have a recurrence score of 35. But if dissected, they would have a much lower score. So these are the types of studies that we did to create how is the pathology reviewed? How do we manually dissect? What needs to always be removed? And in this example, this highlights we always remove biopsy cavities. We then went on and did a series of studies in parallel to pick genes that were associated with breast cancer recurrence and chemotherapy sensitivity. And we essentially did 3 different studies, one with the NSABP, and you can see the other 2 institutions here, in around 400 patient samples that were either node negative or node positive, that received only tamoxifen, or some who received tamoxifen or chemotherapy. And at that time, there was no next generation sequencing. So we started off with 250 genes and asked for each of these studies, and we got these 250 genes from reading the literature on biomarkers in breast cancer, asked which genes are associated with a better outcome and which genes are associated with a worse outcome. And it's from these studies that 21 genes were selected, 16 for cancer, for quantitating cancer biology, and 5 reference genes for a total of 21 genes. And these are the genes. Some of these are estrogen related and are associated with a better outcome. Proliferation are 5 genes, some of which you know, Ki67, others which are involved with anti-apoptosis or cell cycle or atypical mitoses. Like STK15 or aurora kinase, HER2 and GRAB7, genes that were associated with the stroma, stromolysin and cathepsin, and even a macrophage gene here. These genes were most strongly associated with recurrence risk. A patient sample is tested, the levels of their genes are tested, quantitated, and that generates a recurrence score result after their gene expression is normalized with the 5 reference genes. In each of these genes, we measure in triplicate, so 3 different times to ensure reproducibility. Now, we went back on one of our studies recently, SWOG 8814, and we went back and tested those RNA samples and said, using next generation sequencing, where we can sequence every gene in the human genome, will we find more genes, perhaps genes that we could add to the recurrence score? And what we identified, and Kathy Albain presented this at San Antonio in 2014, was we found 568 genes, some genes that were associated with a better biology. And this is showing you an example of genes that regulate transcription that are highly associated. This is 5 times 10 to the negative 7, the p-value, so a point and then 5 or 7 zeros, so strongly associated with outcome. And also many genes that were associated with a bad biology, many of which were proliferation markers or DNA repair markers. But what was interesting is once we put them in a multivariate model and adjusted for the recurrence score, there were only 31 genes that were associated with outcome. The initial 16 genes have been shown, even now with next generation sequencing, to really be very relevant even today when one can sample the entire genome. And if we were going to add genes from these studies to the recurrence score, there would only be a limited number of genes that we could add. Attention to PCR, to dissection, to normalization results in data we presented, first of all, back in 2011, showing 6 years of recurrence score results. And this is showing you on the x-axis the recurrence score result, 2011, 2010, 2009. And you can see that the distribution of recurrence score result is almost identical year after year. And this is the type of analytical validation that results in similarity of results over time. This year, during COVID, we reviewed our experience looking at Needlecore biopsies. And the question during the COVID lockdown was, can we send in Needlecore biopsies? Or do Needlecore biopsies, are they similar to excisions? Or are their recurrence scores systematically higher or systematically lower? So we looked at the million cases, actually 972,000 cases that we had. And there were 131,000 biopsies, 850,000 excisional samples. What we showed is the failure rate is a little higher for a Needlecore biopsy at 5%, 5.5, versus an excisional sample, which is 2.6%. And this is largely due to insufficient tumor tissue. But more importantly, in purple, this shows you the distribution of recurrence scores for Needlecore biopsies. And this in green shows you those from excisional samples. They are not paired samples, but you can see that the distribution of hundreds of thousands of cases are virtually identical. And in fact, their median recurrence score rates are identical. We're not seeing a skew or a bias of the Needlecore systematically having a higher score or a lower score. And when we looked across ER and PR, whether it be biopsy versus non-biopsy or PR or all genes, what we found is that the median expression is very similar between a Needlecore biopsy and an excisional sample. And this, of course, was very important during the pandemic when we virtually saw in the United States patients not being seen for several months, many of whom were being deferred for endocrine therapy. We saw that distribution of Needlecore biopsy go from 13% to around 35% for several months during the lockdown, as the recurrence score was being used for identifying low-risk patients for whom neoadjuvant endocrine therapy would be appropriate during the lockdown. Now, the clinical validation studies that we have done, and I'll quickly just show you B14 and B20 because I know you've seen these, is we worked with the NSABP in 668 patient samples and showed using the recurrence score that we could show amongst those in B14 who were node negative, 50% who had low risk scores and a very low rate of recurrence, distant recurrence of 6.8%, versus those that had intermediate and high risk scores, around 25% of patients. And when we did the same work with the NSABP in the B20 trial, node negative patients who were ER positive who received chemotherapy, again, we saw 50% of patients in the green lines who received chemo in the solid line or in the dotted line, tamoxifen alone, in which in B20, the chemotherapy benefit was seen in that 25% of patients with the red lines who had high risk scores. And this was reflected in showing prediction or this interaction between high risk score and chemotherapy treatment benefit. But there remained, as we segue now and talk about TAILOR-X, it was clear if a patient had a high risk score, there appeared to be large chemo benefit. If they had a low risk score, little or no benefit. But the question was, what about patients with intermediate range scores? And that is what led to the TAILOR-X study design, in which over 10,000 patients, and I know sites in your country participated, over 10,000 patients were tested with the Oncotype test. You can see the range of their patient age, the tumor size, the range of T1 and T2 tumors. And the patients who had scores that were 11 to 25, as I think many of you know, were randomized to endocrine therapy versus chemotherapy, approximately 3,300 patients in both of these arms. And this was a non-inferiority study. And the question was, do the patients who receive endocrine therapy to show whether or not they did worse than patients who received chemotherapy, the standard of care? And the primary endpoint was invasive disease-free survival. You can see the stratification factors: menopausal status, chemotherapy, tumor size, in recurrence score results in groups. I'll quickly highlight a couple of the patient characteristics. Median age in both arms was similar, 55 years of age. Median tumor size, and this is centimeters, 1.5. You can see the range of tumor size here. 13% had less than a centimeter, 63% 1 to 2 centimeters, 24% greater than 2 centimeters. You can see the range of histologic grade, very similar, majority having intermediate or moderate differentiated tumors. And then clinical risk here refers to a modified adjuvant online definition. A patient could be low risk if their tumor was smaller than 1 centimeter in grade 3, smaller than 2 centimeters in grade 2, or smaller than 3 centimeters in grade 1. That was low risk. So any other tumor would have been considered high risk. And you can see by that definition, the adjuvant online, which was also used in the MammaPrint study, MindAct, the 3/4 of patients were considered clinically lower risk. The primary analysis showed that patients who received endocrine therapy alone, here in the gray line, primary endpoint was 9-year invasive disease-free survival, and you can see their risk of the probability of invasive disease-free survival is virtually similar to the patients who received chemotherapy. So this was considered a primary objective that was positive. You can see the secondary endpoint here is the probability of distant recurrence-free interval, or freedom from distant recurrence. And again, no difference between those that received endocrine therapy versus those that received chemotherapy alone. And then you can see in this figure, in the green line, the risk of distant recurrence at 9 years was 3%. The risk of distant recurrence, as I just showed you, for the randomized patients in blue and yellow, virtually lying on top of each other, is 5%. And for high risk, even in spite of chemotherapy, there was a risk of distant recurrence of 13%. What was surprising is that although the clinical factors, tumor size and histologic grade, although there was a trend for poor differentiation or high grade, these factors were not shown to predict chemotherapy benefit. There was an interaction with patient age, in that patients who were less than 50 years of age appeared to derive chemotherapy benefit. And you can see that in this hazard ratio of 1.5, where these confidence intervals don't cross 1. When this was looked at closer in Joe Sperano's presentation, on the plot on the right, he is showing you patients who are greater than 50 years of age, those that receive chemo or no chemo, and you can see virtually no difference is the recurrence score ranges from 11 to 25, and the dotted lines are the confidence intervals. But this was quite different when one looked at the patients, around 2,200 patients who were of age less than 50 years of age. And you can see here in red those patients that received endocrine therapy alone versus those in blue who received chemotherapy. You can see as the recurrence score increases, this chemotherapy benefit increases. And what that showed was that patients who had recurrence score of 11 to 15 who were less than 50 years of age, the curves were overlapping. But from 16 to 20, there was around a 2% chemotherapy benefit, or ovarian suppression. That's the debate. And then patients who had scores of 21 to 25 who were less than 50 years of age showed a chemotherapy benefit of 6.5%. On the other hand, for women greater than 50 years of age, remember the curves overlap. So from 0 to to 25, there was no apparent chemo benefit, and greater than 26 is the presumed chemotherapy benefit. If we look now to a few slides from node-positive breast cancer, in the first 10 years of the recurrence score, we did multiple studies like B14 and B20 using formalin-fixed paraffin-embedded samples because they had been stored by the cooperative groups. We also did a prospective study with the West German study group called Plan B. We did a large registry with Clalit from Israel, which is a large payer in Israel, and had over 700 patients from this registry, and SEER data from the United States. But the most important data were from SWOG 8814, which was a study for postmenopausal patients who were hormone receptor positive. And in this trial, there was the archive samples that we had, around 370 samples. And again, the same question was asked. Let's measure the recurrence score using those samples and see if there is chemotherapy benefit as a function or due to the recurrence score. And what was observed was that patients with low risk scores, and again, this is around 50% of the patient population, did not derive chemotherapy benefit in comparison to those patients who had high risk scores where there was chemotherapy benefit. And again, using a test for interaction, a statistical test, the p-value confirmed the score showed an association with treatment benefit, meaning higher risk patients demonstrated to have chemotherapy benefit. But again, there was a question, not many patients, only 367. And again, a question of, do intermediate patients derive chemotherapy benefit or not? When we looked at 8814 and the smoothed curves in gray is showing you patients who received endocrine therapy alone, and in orange, those that received chemotherapy. And again, right around 25, there appeared to be separation, so chemo benefit. So again, like TAILOR-X, patients were included in responder who had scores from 0 to 25. So a little bit different than TAILOR-X, which was 11 to 25. And again, 5,000 women were tested, 5,000 women were randomized who had scores 0 to 25. You can see there's 2,500 patients who received endocrine therapy versus similar numbers of patients who received chemotherapy. This is the data that Kevin Kalinsky presented at San Antonio this year. And the primary objective was a little different in this study. TAILOR-X was non-inferiority. In SWOG, in the responder study, the primary objective was to determine the effect of chemotherapy, if any, in invasive disease-free survival in women who had node-positive disease in recurrence score results 0 to 25. In terms of race, largely a Caucasian population, but you'll see significant numbers of patients who indicated that they had Hispanic origin. If you look at menopausal status, you can see around 2/3 of patients were postmenopausal, 1/3 were premenopausal. The recurrence score distributions from 0 to 25, from 0 to 13, it was 42%. The remainder had scores 14 to 25. You can see the number of positive lymph nodes. 65% had 1 node positive, 26% 2 nodes, and only 9% 3 nodes positive. Here's the distribution of histologic grade, low, intermediate, and high. The primary analysis was to ask, is there an interaction between the recurrence score and chemotherapy benefit? And what the primary analysis demonstrated was no. From 0 to 25 in all patients, the recurrence score did not predict the relative benefit of chemotherapy. And what does that mean? Well, what that means is there was not, as the score increased from 0 to 25, there was not greater chemotherapy benefit. And so the question is, why is that? Is that because there's no benefit at all from 0 to 25? So in this analysis plan, the first question was prediction. Can we predict chemotherapy benefit from 0 to 25? Since there was no prediction from 0 to 25 in these patients, the next step was to drop the interaction term and now ask about prognosis. Was the score prognostic? And what the primary analysis now demonstrated around prognosis is both both chemotherapy and the recurrence score were prognostic, meaning patients who received chemotherapy had a lower risk of recurrence. Patients with increasingly high recurrence scores had an increasing risk of recurrence. Let's look at this using Kaplan Meier. What this demonstrates for invasive disease-free survival in the overall population, so this is all 5,000 patients, in red are those that received chemoendocrine therapy, and in blue endocrine therapy alone. You can see the number of events, 198 that received chemo and 250 in those that didn't. There was a 1.4% benefit from the addition of chemotherapy. If we now look at patients postmenopausal on the left versus premenopausal on the right, you can see that for the 2/3 of women who were postmenopausal, there was no benefit from the addition of chemotherapy. There was no difference in invasive disease-free survival. If you look at the premenopausal patients, you can see a 5-year invasive disease-free survival of 5.2%. And I've just got a couple of more slides. When we look at distant recurrence, you can see there was a 0.3% difference in distant recurrence for postmenopausal patients, not statistically significant. But for women who were premenopausal, you can see the absolute difference in distant recurrence as a first site was 3%. So for invasive disease-free survival, a difference of 5%. If one looks at distant recurrence alone, 3%. And when we look at postmenopausal patients on the left versus premenopausal patients on the right, if you look at postmenopausal patients for age, grade, tumor size, there was no apparent benefit of chemotherapy. 3 positive nodes, 1 positive node. On the other hand, looking at premenopausal patients, with the exception of women who were 50 or more, all women, irrespective of tumor grade, tumor size, showed chemotherapy benefit. So this is my last summary slide. And Professor Gomez, I tried to move quickly. But in summary, looking at TAILOR-X, which I think you participated in, it's very clear for a woman who's greater than 50 years of age, where there's minimal or no chemotherapy benefit from 0 to 25, whether they are node negative or node positive. For premenopausal patients, in TAILOR-X, women who are less than 50 years of age, there is a chemo benefit that starts to show up at 1.6%, score is 16, and increases as the score increases. And for premenopausal patients, there was a 3% chemotherapy benefit observed, whether a patient had a score of 0 or a score of 25.